Hello everyone. In this video, we are going to study complete plant tissue. For half yearly, I'm making this video. So let's begin. So talking about tissue, what is tissue? The definition of tissue. A tissue is a group of cells that having common origin and are similar, which perform a specific function. So tissues are group of cells or cluster of cells. Now this you can see level of organization is given. Group of cells form tissue, tissues form organs, and organs form organ system form organism. So this is the level of organization in living organism. Moving ahead, we will study the role of tissue. Why do we need tissue? The importance of tissue or the utility of tissue may be asked in your examination. So 3 points I have written. Tissues reduce the work of individual cells. They provide a division of labor. They help to form organs and organ systems. So these are the 3 points you can write. Now study of tissue. Study of tissue, it is known as histology. Whenever you heard the term histo, it means something related to tissue. Now the term tissue was given by Bychet. So Bychet was the scientist or the biologist who coined the term tissue. Mayer was another biologist who elaborated the concept of tissue by studying more about it. Now talking about why do plants and animal tissues are different? Why are we studying plants and animal tissues differently? Why aren't we studying them together? Because plants and animals are two different living organisms. Plants and animals have different key features, different characteristics. Like animals can move, whereas plants cannot. Plants are stationary, whereas animals show locomotion. Plants make their own food, whereas animals do not. They are heterotrophic, whereas plants are autotrophic. Plants tend to grow throughout their life. Plant tends to grow. There is no limit, whereas animals stop growing after a period of time in their life. So this is the basic difference. Now classification of plant tissue, very important, don't forget it. This about 40% of the chapter is this table only. Table help you memorizing things easily. So now plant tissue can be mainly classified into meristematic tissue and permanent tissue. Meristematic tissues have the dividing capacity. So they are dividing tissues. Whereas permanent tissue, as the name suggests, they are permanent. They do not have dividing capacity. Now based on location, meristematic tissue can be divided into apical, lateral, and intercalary meristem. Now permanent tissue can be divided into simple permanent tissue and complex permanent tissue. Now simple permanent tissue, as the name suggests, made up of it has simple structure. It is made up of only one type of cell, whereas complex permanent tissue is made up of more than one type of cell, which makes its structure very complex. Talking about simple permanent tissue, we have 3 main types of simple permanent tissue: parenchyma, chlorenchyma, and sclerenchyma. Parenchyma is the most abundant type of simple permanent tissue. Parenchyma, there are 2 modifications of parenchyma. One is chlorenchyma, and the other is arenchyma. We will study about it. Okay, now this is very important. In your examination, the difference between meristematic and permanent tissues may be asked. So meristematic tissues, as I said, they have dividing capacity. They divide repeatedly throughout their life. Whereas permanent tissue, these meristematic tissue on maturity turn into permanent tissue through the process of differentiation. I think you have studied. So differentiation is the process of gaining permanent fixed shape, size, and function. Specific function. So through the process of differentiation, meristematic tissue matures into permanent tissue. Meristematic tissue have dividing capacity, whereas permanent tissue have lost their dividing capacity. Now cells in meristematic tissue, the cells are very small and isodiametric, means same diameter, whereas permanent tissue, the cells are variable in shape and size, as you have studied. Now intercellular spaces are generally absent, whereas in permanent tissue it can be present or absent. Meristematic tissues have no vacuoles. Why? As you know, because their function is dividing. Their function is reproduction, not storage. Whereas permanent tissue have the function of storage, so they have large vacuoles, and they are also matured cells. So matured plant cells have vacuoles. Now meristematic tissue have thin cell wall. Permanent tissue generally have thick cell wall. It may also have thin cell wall. Now very important, the classification of meristematic tissue. Now meristematic tissue can further be classified into apical meristem, intercalary meristem and lateral meristem based on their location. So the criteria for classification is location. Apical, as the name suggests, it is derived from the word apex. Apex means tips. Apex means tips. So apical meristem are located at the tips of stems and roots, and that is why there are two types of apical meristem. One is SAM, shoot apical meristem, and the other is RAM, root apical meristem. As the name suggests, shoot apical meristem is located as the name suggests, shoot apical meristem is located as the name suggests, shoot apical meristem is located at the shoots and root apical meristem is located at the roots. Now, their main function is growth. They increase the length. They help in elongation. Intercalary meristem. Now, talking about intercalary meristem, as the name suggests, intercalary. So, there are nodes. If this is a plant, and from this point a leaf or another stem arises, then this point is called node. And above the node, near the node, this intercalary meristem is located. So they are located near the node or at the base of internode. If there are two nodes in a plant, there are two nodes, this area would be internode. The space between two nodes is internode. So they are located at the base of internode or they are located near the nodes. Their main function is also elongation, but they help in the elongation of internode, the distance between two nodes. So that is there. And since their main function is growth, and growth means elongation, increasing the length, not girth. So they are known as primary meristem. So this is also important. Primary meristem are apical and intercalary, whereas lateral meristem, also known as secondary meristem, is located at the sides of stem. For example, if I have this plant, so in the cambium, it is also known as cambium meristem. So in the cambium, this lateral meristem is located. It help in increasing the girth. Girth means thickness of the plant. So help in increasing diameter, girth, or you can say thickness. There are many terms. Now this diagram is very important. Very important. Remember this diagram. Location of meristem. So if this is a plant, then this is the shoot tip. So it is shoot apical meristem or simply apical meristem. Now this is node. From here one stem is raised. So this is node. So near that node, the meristem located is intercalary meristem. Now if this is the cambium, so in this stem, trunk, for tree I would use the term trunk, but at this area is the lateral meristem located. It increases the girth or the thickness. So this diagram is very important. Learn it. Now very important, simple tissue and complex tissue. We have to analyze the difference between simple and permanent tissue, complex tissue. Simple tissue, as the name suggests, their organization is very simple. They are made up of only one type of cell, whereas complex tissue is made up of more than one type of cell. The cells may perform similar function, whereas these cells may perform different functions, but generally they also perform similar function. Example you can write as a difference: collenchyma, parenchyma, sclerenchyma, xylem and phloem are the complex tissue. Xylem and phloem together, I'm saying together, they form vascular bundle. Vascular bundle they form. Now this chart is very important, because about 40%, the remaining 40% is covered by this portion, simple permanent tissue. Now parenchyma, collenchyma, sclerenchyma are three simple permanent tissue we have discussed. Now we will study these tissues comparatively. So we are going to have a comparative study of parenchyma, collenchyma and sclerenchyma. Parenchyma is living, collenchyma is also living, sclerenchyma is dead. This is the difference, first difference we have got. Now in parenchyma, intercellular space is present. The space between two parenchymatous cell is present. In collenchyma, the space may be absent or present, not confirmed. But in sclerenchyma, the space is absent. These are compactly packed cells. Now cell wall. Parenchyma cell wall is very thin. The parenchyma cell wall is very thin and made up of cellulose, whereas collenchyma cell wall is thick from the corners only. For example, if this is a cell, if this is a cell, if this is a cell, then the what I'm saying, the corners would be thick. The corners would be thick because of the deposition of pectin and hemicellulose. So pectin, because of the deposition of pectin and hemicellulose, so it is made up of pectin and hemicellulose. Now sclerenchyma is uniformly thick. This is the cell, so it is uniformly thick, because of the presence of lignin. So cell wall in parenchyma, the cell wall is made up of cellulose. Collenchyma, it is made up of pectin and hemicellulose, and in sclerenchyma the cell wall is made up of lignin. In parenchyma the cell wall is thin, sclerenchyma cell wall is thick, collenchyma is intermediate. Okay, now function, very important. Function, parenchyma, whenever you heard the term parenchyma, salt storage. So the function of parenchyma is food storage and photosynthesis sometimes, but main function is food storage. The function of collenchyma is to produce flexibility. I think you have seen plants which have stems that are much flexible, like the tendrils and the stems of climbers and creepers. So they have collenchyma, so it provides flexibility and mechanical support, and the sclerenchyma provides stiffness, hardness, strength. So support, stiffness, hardness is the function of sclerenchyma. Now location, where they are located. Parenchyma is located almost in every part of the cell. As I have said, it is the most abundant cell, most abundant permanent cell, permanent tissue, sorry. Now collenchyma is present in the hypodermis, petioles of leaves and stems and tendrils. Collenchyma is present in hypodermis, petioles of leaves and stems, and in tendrils. Whereas the sclerenchyma is present in the husk of coconut, very hard, stems, roots, the parts which are very hard. So there sclerenchyma is present. So sclerenchyma is present there. Now, talking about the diagram, very visual understanding is also important. See, intercellular spaces are present in parenchyma. They have thin cell wall, and this you can see. Now in collenchyma, this is the longitudinal view, not so important. Collenchyma, the intercellular spaces are maybe absent or present, do not concern about that, but the cell wall is thick from the corners at this point, at this point, at this point. So it is thick from the corners. It is thick from the corners. Talking about sclerenchyma, in the sclerenchyma, the cell wall is uniformly thick. You can see sometimes the cell wall is so much uniformly thick that the intercellular spaces is almost negligible. Now, modification of parenchyma, very important. It is also, parenchyma can be modified as chlorenchyma and arenchyma. Chlorenchyma, a very basic. Chlorenchyma, as the name suggests, it is related to chloroplast. So the parenchyma that contain chloroplast are known as chlorenchyma. What is their function? Food synthesis, that is photosynthesis. So they perform photosynthesis. Whereas arenchyma, as the name suggests, air. So they have air cavities or air sacs which help them to float on water. So they provide buoyancy, float on water because of the presence of air sacs. So this you can see, this is chlorenchyma containing chlorophyll, cells which contain chlorophyll, and this chlorophyll and chloroplast. And this is the arenchyma. They have air spaces. These are the cells. These are cells, and these cells have some air spaces. Now the last topic, not the last topic, the second last topic, that is complex permanent tissue. Name suggests they are made up of more than one type of cell, xylem and phloem. The difference may be asked. As you have studied from the lower classes, xylem help in the transportation of water, known as ascent of sap. the water containing mineral is known as sap. So the xylem's main function is transport of water and mineral, that is sap, that is known as transportation of water. And phloem, it helps in the translocation of food, and the process is known as translocation. It is transportation, it is translocation. Xylem is unidirectional, that is it flows only from roots to the stem, from down to the up, whereas phloem is bidirectional, means from the leaves. If this is a leaf, so from the leaf the food will go down also, from the leaf the food will go up also. So it is bidirectional, both the direction up and down, both the directions up and down, whereas it is only up. Now xylem is mostly made up of dead cells. There is one living cell, whereas phloem is mostly made up of living cell. There is one dead cell. Now this slide is very important. Elements of xylem and elements of phloem. Elements means the different cells which constitute xylem. So tracheids, vessels, xylem fibers and xylem parenchyma are the elements of xylem, whereas sieve tubes, companion cells, phloem parenchyma and phloem fibers are the elements of phloem. See, tracheids and vessels, they together function for the conduction of water. So their main function is water conduction. So they form main water conducting channel, tracheids and vessels. And xylem fibers, whenever you heard the term fiber, then it is supportive in nature. Its main function is support. If you heard the term xylem parenchyma, parenchyma storage of food. Now these three, tracheids, vessels and xylem fibers are dead, whereas xylem parenchyma is living. Now sieve tubes in phloem, if you see, sieve tubes and companion cells together help in translocation of food. Actually the main transport is done by sieve tube, but companion cell help the sieve tube in the translocation of food. Now phloem parenchyma is also there. Whenever you heard the term parenchyma, it is a storage of food. And now phloem fibers, whenever you heard the term fibers, fibers means what? Dead. Dead, and they help in support and they provide strength. So these sieve tubes, companion cells and phloem parenchyma. Parenchyma is living, so these are living. And phloem fibers, fiber is dead. So for remembering the trick which one is dead and for remembering which one is dead and which one is not in xylem and phloem, let me tell you a trick. See, in xylem the parenchyma is only living, others are dead, whereas in phloem fiber is only dead, others are living. This is the basic clear difference. Now protective tissue, very important. Last topic, protective tissue. Protective tissue are two types, epidermis and cork. Epidermis and cork. Epidermis is the outermost layer of cells. Many cells together form this epidermis. Below epidermis we have this hypodermis. So now epidermis is living flat cells made up of living flat cells. Intercellular spaces are absent. What is the function? As the name suggests, protective tissues. So their main function is protection. Protection is the main function of epidermis. Cork also perform the function of protection only. Cork is known as phellum also. So cork is made up of dead cell, whereas this is made up of living cells. One difference. It has intercellular spaces absent. It also has intercellular spaces absent. Now cell wall is thick and made up of suberin. It has suberin, a kind of chemical that is present in the cell wall. Suberin basically is waterproof. What I'm saying? Yeah, no. Suberin is basically, waterproof. No, suberin is not waterproof. Sorry, this cuticle. It has, in desert plants, the epidermis has a layer of wax known as cuticle made up of cutine. Cutine is a chemical. So this layer is a wax layer of cutine, also known as cuticle. This help in preventing the loss of water from the plant. That is, the wax layer of cutine is waterproof. So they do not allow water to transpirate from the inside of the cell to the outside. That means transpiration, and it has some some openings called stomata containing guard cells. Stomata help in the exchange of gases and transpiration of water also. So this is their guard cells, help in the opening and closing of stomata. These are guard cells. These are guard cells. that is just, and in bark of trees you can see this cork present. What happens when the tree, when the plant gets old, the inner cells form cork, and the cork tears this epidermis and come out, emerge as cork. So this is just there. And it has an outer chemical substance. It has an outer chemical substance called suberin.